All right, good morning, everybody. Good
morning.
Like a treat. Puppy shot. Isn't she cute? Okay, let's get the chat up. Share screen. All right, so before we start, because we got through a lot last Friday, does anybody have any questions about assignments, exams, grading, course, etc. before we get going? Feel free to type them up in the chat as well, wherever the chat's gone. Dun, dun. Also, if you don't have questions, that's fine as well. But in case you do. All right, where are we at? Oh, I'm sorry, Monday morning blues. I want to get out of bed this morning. That was my first thought that went through my mind as it woke up. Good question, Gina. Yes, I think would be a good prep for the class. Shouldn't take you too long, about a half hour maybe. Also, because this is the first class, so it's a bit weird, as you can imagine. But for subsequent classes, what I would also suggest you do is, after reading through your notes and kind of doing work and homeworks and things like that, after previous classes, write down a list of questions, right? Stuff that you don't get, things that don't make complete sense, stuff you want clarifying, because that's part of the purpose of what we're doing in these sessions, right? It's not just to go through the PowerPoints. And so then you can bring that to class, and at the beginning of class, if anyone has any questions, we can go through those. Even if it's a couple of PowerPoints ago. If it's prior to the previous exam, then that's interesting, but not as useful. But yeah, use that as part of your kind of revision and study for the material. All right, generally speaking, the more you put into this class, the more you get out of it, is a good way of thinking about it. Okay, so essentially what I want to do today is obviously, field any questions that anybody still has, but also kind of go through a basic outline of what we'll be studying in terms of genetics. Start getting you some words, right? Some language, because we're going to be using a specific language in this course, right? Obviously it's English, because that's one that we all share, but we'll be using a lot of very specific terms that have to be used very carefully, right? There's a lot of precision to scientific language. You have to be very careful about what you say. Classic example is that, people always say, oh, there's a significant difference between these two things. I was like, well, did you do the statistical analysis? No, then you can't say it's significant, right? Because in biology, or probably other disciplines too, significant means something very specific, right? So you have to be careful about the words you use. And so part of what we're going to be doing is giving you that vocabulary so that you can talk genetics, basically, right? So that's another thing that's worth taking down as you go through this course. Okay, so kind of in general, when we talk about genetics, we're talking about two different things, right? One is the underlying material, DNA, RNA, protein, mostly DNA and RNA, and how that varies, like within an organism, within a cell, but also between taxa, right? So between like us and chimpanzees, the whole 99.9% kind of genetic similarity between us and chimpanzees. What do we mean when we talk about that, right? What does that actually mean functionally, right? So that's one part. That's kind of like the nuts and bolts, essentially, right? Of the underpinnings, the mechanism. And then the other part that we'll be looking at is, what do you see, right? What is the expression of that information? And in that, the word we use for that is the phenotype, right? So that's a beautiful dog, Jessica. I don't know what dog is it?
He's an Anatolian Shepherd.
Oh,
He's upset that I'm locking him in the office.
oh, fair enough. Right. So what you see, the phenotype is quite often what people most frequently refer to, right? And that's really the expression of the underlying kind of DNA and gene expression, so on and so forth. But also, how do those traits, right? Hair color, skin color, nose shape, height, disease, right? How are those inherited, right? And so those are the kind of the two big parts of what makes up genetics, right? And obviously, as time goes on, how we interact with that, changes, right? Used to be there was just, gene by gene at a time. Now we're looking at, entire genomes, right? What is it called? The 500,000 haplotypes experiment. There's a thing going on in the UK where they're just sequencing 500,000 human genomes, right? And so they can have this huge data set by which they can look for correlations between a particular trait, such as high blood pressure, let's say, and see if they can figure out from that huge data set what's the underlying mechanism, right? So kind of as our abilities get bigger, the questions get bigger and more complex with it. That's kind of in many ways, one of the most exciting aspects of genetics and biology at the moment. And so if we were kind of to, slice the pie a little bit thinner, essentially, in genetics, we can think of it in one of three ways, right? And these are not hard borders, right? As you'll find with most biology, it's a very kind of human artifact. We want to divvy things up. I mean, that's what taxonomy is essentially in many ways, right? Like this little group, and they have this little group. And those are very arbitrary kind of lines in the sand. They don't really, it's not that they don't exist, but they're not hard and fast, right? And so in genetics, we have the same thing. We have like the nuts and bolts that we were talking about earlier, generally refer to that as molecular genetics, right? And so that's really where a lot of my research has and is taken place, right? So I'm really a molecular geneticist. It's kind of how I view myself. How do these pathways work? How do different things interact, right? So when you go up one scale, so that molecular genetics you can consider at the level of the cell, right? You can consider tissue, even consider at the organismal level as well, right? But when you go up a level, and you can think about this in terms of scale as well, right? It's another neat way of looking at it. When you go up a level, now we have, well, how does that information get passed from generation to generation, right? How do those traits get expressed, right? And this is where we start talking about dominance and recessive. I always like talking about my own family because obviously it's fun. So one of the kind of dominant traits which is passed down through my mother's lineage, I guess you could say, into me and my brother is kind of a biggish nose and hairy toes, right? Obviously, hairy toes is a hard one to see in family pictures, but it's something that's gone down through every single generation on my mother's side. So that's cool. I mean, like, why? I have absolutely no idea. It's kind of still a bit of a mystery. But those are really what the things we're thinking of when we talk about transmission genetics, right? How does that information get transmitted from one generation to the next? So we're still talking about individuals, but now we're talking about generations of individuals. Now, obviously, if you continue that on long enough and look at a big enough sample size, population essentially, now you can start to see those traits and those alleles, right? The versions of genes which control different phenotypes. You can see how that changes within that population over time, right? As new people come in, you know, as people move in San Antonio on a daily basis, right? What gene variants, what alleles are they bringing in? Well, it depends on where they come from, right? If they come from, you know, El Paso or Austin, probably nothing very different to what we already have. You know, if they're coming from Alaska or Vermont, then almost certainly they'll be bringing in something new. And so how does that change over time? And if you look at it over a long enough period of time, and if there is sufficient selective pressure, whatever that might be, extreme heat, right? Crazy humidity, air conditioning, I don't know. Then you can see that change over time, over evolutionary time. And then maybe you'll start to see some kind of speciation, right? And so really, evolutionary genetics is population genetics over time, right? That's kind of the way I always think about it. We're not going to be covering evolutionary genetics in this course because that's going to be covered in considerable depth during evolution, right? Which is kind of a partner course to genetics, but it's the next one along. Ooh. I should get in the habit of having a coffee before class, I think. Right, and so that's typically you can take that either directly after genetics or you'll take micro and then you'll take evolution. Depends on how your degree plans kind of laid out. And most people that study genetics look at more than one of those things. So me personally, as I can talk about myself with confidence, I'm a molecular geneticist, but a lot of the research I do relies on the principles of transmission genetics. Right, so if I want to get two different alleles together for different genes, then I'm going to have to do a genetic cross, which you'll be doing in lab with fruit flies, which is super neat. I need to do a genetic cross, and so I need to figure out, you know, how many worms do I need to look at to be able to get what I want, and really that's down to probability and transmission genetics. Okay, so just a little bit of history, which I try and keep it small because I'm not. I like history, but it's not. Essentially, science is a forward-looking thing, right? We're always looking forward to what is next, not what has been before. But the crazy thing about genetics is it's a very new science, right? Really, it only took off in probably from the 60s onwards, mid 50s, after the these two guys, Maurice Wilkin and Rosalind Franklin, together discovered the structure of DNA, this whole double helix deal, right? But really, like, things didn't actually properly take off until probably the late 70s, early 80s, with the advent of PCR. So PCR stands, if you don't know already, polymerase and that's basically about amplifying DNA. And so genetics actually as we know it now didn't really kick off until that became a viable tool. And that was really in the 80s with the advent of the thermal cycler, the machine which allows you to do PCR really quickly. And then after that, you know, essentially that's what led to the human genome sequence and kind of what we're now familiar with in the kinds of, you know, personal genetics and stuff that we have now. Right, so really you're talking about 40 years, which isn't a very long period of time. All right, so not much left of this one. We'll probably move on to the next PowerPoint in a little bit. Part of what we do at the start of the course is just give you a refresher, essentially, or refresher even, in some of the stuff that most of you probably already know, but it doesn't hurt just to go over it again. Same deal with the homeworks, essentially, right? And so when we talk about DNA, and really DNA is, it's the hereditary material, unless you're an RNA virus, in which case you're not technically alive, right? So all life, however you want to define that, uses DNA as the store of its information. And I'm purposely excluding RNA and viruses from that. Right, and so DNA is quite an astonishing molecule, right? It has a really cool structure. It's also very, very durable, right? So you're able to extract it from, ancient Homo erectus or Australopithecus bones, right? And these are between tens of thousands and hundreds of thousands of years old. And you can still get DNA out of those bones, which is just astonishing, right? You can even go and find yourself a frozen dude. Let's see if I can remember how to spell his name. There we go. No, I couldn't remember how to spell his name. There you go. This guy looks like he could do with a bit of a shave and a wash. Actually, he's in a pretty bad condition. He was spat out by a glacier in Austria, I think. He's about 4,000 years old. Where's the picture of the, oh, there we go. This is a particularly fun picture. So he got shot in the back by an arrow, which I thought was kind of rude. Don't know why. He was wandering over a glacier. Maybe someone didn't like him for it. But anyway, they're able to get the DNA out of this guy and basically kind of plug him into his kind of genetic environment. Like, where's he from? And they actually managed to kind of isolate him down to a particular village in Austria, in the Alps. So isn't that kind of crazy? I think that's super awesome. Anyway, also it's the kind of principle behind a lot of forensic science too, right? This stuff hangs around for a long time. It's a very durable structure. And part of that is this double helix, right? So essentially the information, right? Like the fingers on my hands, essentially, the bases of DNA is on the inside, right? And is protected by this backbone, right? This phosphate sugar backbone, which you can see going down here, which keeps all that information inside it and kind of allows it to kind of wrap up in a fairly small structure, which is very durable, right? Doesn't get attacked very easily by, free radicals and stuff like that. It has a right-handed twist. Something you'll also notice is that it doesn't have an even twist, right? So this isn't like you get a twist tie off a loaf of bread and you just twist it together, right? It's not an equal twist. And so DNA also has what I like to call topography, like a scenery in a sense, right? In that it has this, what's called a narrow groove, and we'll get to this later as well, or minor groove, and this major groove where the strands are the twist is a little bit further apart. That's also an important feature of DNA, not just its sequence, but also its structure too. And it's anti-parallel, right? So if I'm going to just zoom in a bit here, so it makes it a little easier for you guys to see. I think you can all see that. Yeah. Sorry, I often have to move things around because stuff on the PowerPoint's hidden by you lot. Right, so you'll see on the left, right, we've got this backbone, phosphate sugar, phosphate sugar, phosphate sugar backbone. That's why it's called the phosphate sugar backbone. And it's going from 5' at the top to 3' at the bottom. We'll get to what that means later, right? Just keep it in mind for now. You'll see the other strand goes the opposite direction. So one strand is going down, the other strand is going up. We call this an anti-parallel structure. So DNA is a double-stranded, right-handed anti-parallel helix, or right-handed anti-parallel double helix, whichever way you want to put it. And this anti-parallel nature is a very important part of DNA structure, which will have a big impact on a lot of stuff that we cover later. So one thing I try and do, because I've taught this course a bunch of times, and so I know what's coming up, and so I try and give you some, my grandmother used to call them worm in your ear. I don't know what the kind of modern equivalent would be, but like, come on, just put that little thought in your head, and then later when we come across it, you'll be like, oh, right, you know, so you kind of already have a little bit of inkling of what's coming. Right, so this anti-parallel structure is really, really important for DNA replication and for gene expression. Oh, good question, Stephanie. Yeah, part of the reason for how important that is that you can only add new nucleotides to the 3' end of both DNA and RNA. So yeah, you did get the right general idea, just the other way around. And so because of that, it means you can only synthesize DNA and RNA in one direction. Also means that you can have information going this way on DNA and information going the other way, right? So you can actually have genes which overlap. You can have information on both strands. But again, we'll get to that later on. You already know there are 4 bases: adenine, guanine, thymine, cytosine. And what I always have to be careful with terminology. Technically, the base is, pardon me, this part here, right? It's a phosphate, a sugar, and a nitrogenous base. So a nucleotide equals a phosphate, sugar, and nitrogenous base, if I can spell that right. I try and type this stuff out because this is also a record which will go up on Blackboard, right? So whenever I introduce new terminology and stuff, or things that I think are important, I'll put them in the chat. So just in case you don't have time to write them down, or you want to refer back to it later, you have that later on. But kind of, I can't write, how do you spell colloquially? Yeah, I don't know how to spell colloquially, not easily, at least, without thinking about it. Informally, I guess. But quite often, you know, we refer to bases as being the whole thing, right? So formally, a nucleotide is this phosphate, sugar, and nitrogenous base, right? Like the one you can see at the bottom down here. And the base is technically, precisely, just this helical carbon and nitrogen structure here sticking out in between. But colloquially or informally, we often say, well, you know, we've got this base here and this base here. We often call the whole thing a base or base pair, given that we're talking about DNA. So quite often, if I talk about size in terms of DNA, I'll give you a number in base pairs, right? That's how many of these nucleotides there are in that particular bit of information. So sometimes there's a little bit of potential for confusion in terminology based on, you know, the formal definition and what we use on a day-to-day basis. And also those bases, those nitrogenous bases, base pair, right? And they will only base pair in a very specific manner. This is super important. Adenines will only base pair with thymines, and guanines will only ever base pair with cytosines, right? In addition to that, you'll only ever have a pyrimidine. So if I can spell that right. And that's a one ring base. That would be a thymine. Hey, quit, stop auto correcting me. Now let me do this a different way. And so the easy way to remember which bases those are is that the ones that have a Y in them are pyrimidines. So thymine and cytosine are pyrimidines. And I have lots of really kind of kooky and weird ways of remembering stuff because, you know, I'm not a memory genius. But my way of remembering what pyrimidines are is that I imagine a small whiny base going, why, oh, why am I such a small base? It's like sob sob. Now, obviously that's a little bit silly, but you know, whatever works, and that works for me. So pyrimidines are the small bases. Purines are the larger ones. And so you'll only ever get a pyrimidine and a purine base pair. And that's based on size. So essentially, if you look at all the way down here in the middle, if you count up the rings, you only ever have 3. You'll have a pyrimidine, which is 1 ring. You'll have a purine, which is 2 rings. If you had a purine and a purine, which will be 4 in total, the DNA backbone would actually bulge outwards. And similarly, if you had just pyrimidine and pyrimidine, it would kind of dip inwards. And those structural changes are what are detected by the DNA repair mechanism, or one of them at least, which we'll come across a lot later. Now, can anybody remember what these dashed lines represent? What sticks bases together? What holds the 2 strands of DNA together? Can anyone remember what type of bonds those are? Hey, Sierra, wow, that was quick. Yeah, so hydrogen bonds. And can you notice a difference between the T's and the A's and the G's and the C's? If you look at that. What's the difference between the number of bonds? Super awesome, right? So A's and T's will always have 2 hydrogen bonds. And that's the other part of this complementary base pairing. So one thing which forces the right bases next to each other in terms of DNA replication, for example, is the size deal. You can only have a purine with a pyrimidine. What makes sure that the correct purine is next to the correct pyrimidine is the number of bonds. And that comes down to the actual molecular structure of the nitrogenous base itself, which we'll get to later. You'll see why that works the way it does. But this is a super, super important part of DNA replication. And essentially, it's why you're not a gibbering jelly mass of undifferentiated cells, right? Because every time your DNA replicates, it has to do so very accurately. And it's also how we're able to turn the information in DNA into messenger RNA, which can then be translated into that intermediate. Right, so there's a lot of precision needed in terms of DNA and RNA. And essentially, you know, once you know something about DNA, you can understand that it can be copied, right? Again, using that complementary base pairing and extension that we talked about a little bit earlier, right? Information is encoded by what bases you have, right? There are only 4 different bases, but amazingly, we can use those 4 bases to encode. Oh, here's another question. Approximately how many genes do you think we have as a human? Just a rough guess. Anybody know? It's more than I can hold up in my hands, right? Anybody got a rough guess? That's a good guess. Let's go a bit higher. Think about how many different processes. Yeah, that's a good guess. Let's go higher still. Get in there, Nancy. Let's double that, right? Humans have about 20, that's right, Melanie, about 20,000 genes, give or take. Well, actually, Caitlin, that's a cool way of thinking about it. So actually, we can encode, so I don't know enough about computers to do a very good job of answering that question. But the cool thing about how we get to those 20,000 genes is we've got 4 bases, right? And we read those 4 bases in words of 3. They're called codons, right? And as such, we can encode 3 to the power of 4. Yeah, I think that's right. Or is it 4 to the power of 3? I think it's 3 to the power of 4. I'm not a very good mathematician. Different bits of information in terms of words, and those are the amino acids, right? So we can potentially encode 64 different amino acids, even though we only have 20. But each of those genes consists of a sequence of potentially thousands of different amino acids, right? And so the crazy thing about, you know, genetics is that we have 4 bits of information, A, C, G, and T. And we can use those 4 bits of information to encode around 20,000 different genes, right? Which, you know, code proteins that do all kinds of different stuff. You know, something else that's also really funky? Guess how many genes a soil nematode, what I work on for my research, how many genes they have? So these are about, kind of about that long, about a millimeter long, 959 cells. How many genes does a soil nematode have? First organism to have its genome sequenced as well. Now, give you, it's actually a bit of a trick question. They also have 20,000 genes, give or take. So in terms of genetic complexity, in terms of gene number, at least, we have the same number of genes as an invertebrate with 959 cells, which I always think is pretty cool. right? So you can get a hint of how complex genetics is. Yeah. Hey, I didn't know you could do emojis. That's cool. I just got a smartphone for the first time. And neat. And I've just been spamming my family with gifts and stuff constantly. It's like I can annoy them from a distance. Don't even have to be there in person anymore. All right. Anyway, let's not get distracted. Also, that information is not static, right? I don't mean that it moves around, but over generation, that information can potentially change, right? And as such, those changes may be inherited in a way that leads to, you know, divergence of populations and speciation. So what else is there in here? Yeah, a little bit on genomics. We're not going to really do much on genomics because it's a whole nother kettle of fish. But really what we mean about genomics is not looking at each individual gene. Now we look at the whole genome. How does that change between different species, right? Can they explain some of why those species are different? Can we look at, and you know, can we, you know, sequence my genome and try and figure out why I have a particular disease, right? Or even more interestingly, how am I going to respond to a treatment for a particular disease, right? That's actually one of the hottest topics in medicine right now, is this idea of personalized genomics, right? So quite often now, if you are unfortunate enough to have cancer, this has actually happened to niece of my wife's. She had kidney cancer. One of the things that they did was they did a genome scan, right, to see whether this was a spontaneous cancer or if it was a inherited form of cancer, right? Because that informs how they're going to treat it. They may, I don't actually know, but they may have even looked for particular markers for drug resistance, because an awful lot of people, if you give them the chemotherapy, they don't respond, right? Because they either metabolize that drug or the cancer that they have is resistant to it. So genomics is arguably the kind of the biggest thing happening really in modern science, I think, you know, at least in modern biology. And that's really sprung from the whole genome sequencing of the human genome, which was published nearly 20 years ago now. So it's taken a while, but it's getting there. Okay, so that's pretty much it for this particular PowerPoint. Does anybody have any questions? Let me just get this back up to the beginning. Yeah, get out of it. I do like that that emoji, Caitlin. I think that's super awesome. I don't know how that's going to translate to a text file though. So genome is your whole genetic information, really is the best way of putting it. So that's not just, well, it includes all of the protein coding genes, however many of those are, and it includes all of the rest of the DNA that you have within your cells. So technically, if we're being accurate, it can be split into your nuclear genome, and so this is the DNA that you inherit from both parents, and also your mitochondrial genome, if I can spell that right. And that you only get from your mother. So you can actually trace your genetic lineage through the matrilineal line back thousands and tens of thousands of years, because you only get mitochondria from your mother because the sperm doesn't contain any. Sperm just contains DNA, basically. So those two together are the sum of the genetic information that you possess. And so when we talk about genomics, we're really comparing those, right? So how does my genome compare to your genome, right? What differences are there? Those differences are both because obviously we have different parents, at least I'm pretty sure we do, but also because, you know, my genetic kind of ancestry is Northern European by and large, Scotland, Germany, hopefully no French, but anyway, sorry. That's something that I know Americans love the French. British people are basically raised to hate the French. I don't know why. They're really nice people. It's just like this kind of ancestral thing, I guess. Right. And so if you compare my genome to genome of someone who kind of grew up around, you know, the border, Mexico, Texas, California, something like that, even though we're going to be largely identical, right? The differences are really, really, really small. Those differences will be the result of whatever population your ancestors came from versus a different population that my ancestors came from. You know, so that's one thing that makes it really interesting. That's also one thing that makes this whole personal genomics really interesting too, because although one has to be very, very careful talking about race in terms of biology, people who have a genetic lineage from different areas of the world, right? Because race is a bit of a, it doesn't really have a biological basis, right? But people who come from different parts of the world have very different kind of genetic ancestry in a sense, right? And the alleles that they have inherited as a result of that ancestry may give them a greater sensitivity or resistance to particular diseases and treatments, right? And so you'll tend to find, if you can kind of tease out the socioeconomic aspect of it, that people of different areas respond differently to different treatments, right? So it's a really, really interesting, it's a very contentious kind of area of medicine, right? Because you can end up causing more problems than you solve if you put race at the forefront of how you treat people. But it's also part of, you know, who you are genetically as well. So it's cool. Going on a bit of a tangent there. Okay, so just to kind of ease into the next lecture, and sometimes we don't fully kind of keep to the schedule. It's more of a guide than it is a prescription, depends on questions and tangents and stuff. But sometimes we'll start a PowerPoint early, sometimes we'll go over into the next lesson, things like that. It's not too big of a deal. There's space in the schedule for that kind of flexibility. Get ahead of it. My chat box keeps wanting to expand to fill my whole screen. Here we go. And so what we're going to do in this is really kind of get across the, hey, sorry, this dog, if you can see her, there she is. She's kind of growling in her sleep for some reason. And she's still sleeping. Took her for a walk and completely tuckered her out before class, which is kind of funny. Anyway, so what we're going to do in this PowerPoint is really recap what you've learned in GenBio 1. Structure of DNA, the principles of gene expression, which is really how we go from DNA to something functional, right? Because DNA doesn't do anything, right? It is a store of information only. And so if you want to do anything with that information, you have to turn it into something else. And that something else is RNA or protein, right? And then we'll talk a little bit about the actual process of transcription and translation, which make up gene expression. And then we'll talk a little bit about different kinds of mutations, right? For example, what I was talking about with my niece-in-law's kidney cancer, right? Is it a spontaneous mutation or is it inherited? And that's pretty much it. So we'll leave that for, because there's only a few minutes left, there's no point kind of diving into it now. We'll leave that for next class. So have a look through that. At the moment, we're still in the kind of the sub-exponential phase of our course. Soon we'll kind of start taking off and we'll really get into the weeds. But right now we're still just recapping, making sure everybody's on the same page. And that's pretty much it for me. So yeah, sometimes we finish a little bit early. I've never had anyone complain about that yet, to be honest. Question about lab and lab coats. You do not need a lab coat for your first lab. Right, so, but make sure you do have one for your second lab. You will need one for them, and we can't rent you one. So if you don't have one, it's kind of awkward. We'll have to basically send you away until you can find one, right, from a friend or something like that. So yeah, bring a lab coat for not this week or next week, for the lab after that. Also as well, the registrars have been playing silly buggers again with the course numbers. And so for my sections, oh, that's very generous of you, Melody. For my sections, the odd sections are meeting first, the even sections are meeting second. Right, so the odd sections will be attending lab this week. So if you're 01 or 03, you'll be meeting this week. If you're 02, 04, you'll be meeting next week. That doesn't necessarily apply for Mr. Penny or Mr. Chan's, Mr. Penny or Ms. Chan's class. I think Ms. Chan actually has it the other way around for whatever reason. I don't know why. Okay, so refer back to them. For they're essentially the horse's mouth when it comes to lab. All right, everybody. I'll leave this up for a couple of minutes if people need to sort out lab coat stuff before anything else. Will you take to lab tomorrow yourself? Is pretty much it. If you have a device which you can have the lab handout on, take that. You can print it out if you wish to, but I'd actually recommend, if you can, if you have like a tablet or a laptop, bring that, because sometimes you can actually do some of that in lab, because the actual assignments are all on Blackboard. So, but you don't actually need to do that. So you can just bring a printout. But you need to bring something that has the lab handout on it, because that's what you refer to in lab. Cool. Yes, you can use your laptops in lab. There's no kind of contamination issue with the labs that we do that would prevent you from doing that. And there's things you can plug chargers into and stuff like that too if you need to. Cool. All right, everybody. Have a lovely day, and I shall see you either tomorrow or on Wednesday. All right, take care.